Hey guys, welcome back to One Stop Biology. So guys, today we are going to basically study about the factors affecting enzyme activity. So if you remember, if you have gone through the previous video, we have basically almost covered the entire chapter of biomolecules, which is chapter 9 of NCERT Class 11th. And we are right now running the NCERT series where we are covering the syllabus as per what is given in NCERT. So guys, we have already covered a lot about different type of biomolecules, biomacromolecules, biomicromolecules, right? And in the last video we studied about enzyme. So we studied the definition, we studied how the enzyme works, how the chemical reaction happens, what is the concept of activation and energy, and then we also saw what is the nature of enzyme action and what exactly happens when we talk about the enzyme action, right? So today we are going to discuss factors affecting enzyme activity, where we'll see what all things actually affect the enzyme, the working of an enzyme. So let's start with that. So basically guys, the activity of an enzyme can be affected by a change in a lot of factors. Like say if something happens that alters the tertiary structure of the enzyme, the protein, it will basically start malfunctioning. So it will stop working or it will not function properly, right? So basically there are a lot of functions or a lot of different factors which affect the activity of the enzyme. So this basically guys includes temperature, pH. Say if there is a change in concentration of of substrate, so change in substrate concentration, or if there is some change in binding of specific chemicals. So basically change in binding of specific chemicals, right? So guys, what we are going to do is we are going to discuss all these factors individually, and we'll start with temperature and pH. We'll start with temperature and pH. So basically guys, as we discussed in the last video as well, that if the temperature increases, the enzyme stops working, right? So enzymes generally function in they basically function in a very narrow range of temperature and pH. Remember that enzyme works in a narrow range of temperature and pH, and this basically is the temperature or the pH at which it works is known as optimum temperature or optimum pH, right? So basically below or above. So remember, below this or above this optimum temperature and pH, the activity of the enzyme declines, right? So the activity of enzyme declines. Now remember one more thing guys, that low temperature, if the temperature is low, it is below the optimum temperature, that preserves the enzyme in a temporarily inactive state. But if the temperature increases, that means temperature higher than the optimum temperature destroys enzymatic activity, as enzymes are basically made of proteins, and these proteins are denatured by heat, right? So remember this statement guys, that low temperature, This means temperature below optimum temperature. What it does is it preserves enzyme temporarily, right? In an basically in inactive state. It was not worked, but yes, it is preserved. But if the temperature is high, that means higher than the optimum temperature, this destroys enzymatic activity. So this does what? This destroys enzymatic activity, right? Because the protein basically is denatured. So again, guys, here if you see this diagram, you will see that basically what is happening here is that this basically is the pH and this is the enzymatic. This is the enzymatic activity. Okay, so say till a time the activity increases, and then after this threshold it starts decreasing in case of pH. Again, that same happens in case of temperature as well, that the enzyme activity increases, and then after a particular temperature it starts decreasing, right? And we'll see the graph of concentrate of substance as well. So once we start off with concentration of substrate. So basically, guys, what happens is that with increase in substrate concentration, the velocity of enzyme reaction, rises at first. So here if you see, this is the substrate and this is the velocity of reaction. This is the velocity of reaction, V. Right. So what is happening is that till some time, what happens is with increase in substrate, the velocity of enzymatic reaction rises. But then it reaches to maximum velocity. So basically this is V max. So there's a point when it reaches to the maximum velocity, which is not exceeded with rise in concentration of substance. So after reaching this V max, it becomes stagnant and it basically stops increasing even if you increase the concentration of substrate, right? So basically the activity of an enzyme is also sensitive to the presence of specific chemical that bind to the enzyme. Say if the binding of chemical shuts of enzyme activity, that process is known as inhibition. That process is known as what? That process is known as inhibition. This is happening when there is a chemical which binds to the enzyme and it shuts its activity. And such chemicals are known as inhibitor. Such chemicals are known as inhibitor. Okay. Now when the inhibitor slowly resembles the substrate in its molecular structure and inhibits the activity of the enzyme. So sometimes what happens is that this inhibitor, it very closely resembles the structure of the substrate, and what happens is it inhibits the activity of the enzyme. It is also known as competitive inhibitor. So you'll have to remember all these words. So when a chemical substance, right? When there is a substance, chemical substance, which stops the activity of enzyme, it is known as inhibitor, and that process is known as inhibition. Now sometimes this inhibitor resembles the substrate which is going to bind with the enzyme basically. So it binds with the enzyme and it inhibits the activity of the enzyme. That is known as competitive inhibitor. Right. Now what happens is that basically due to close structural similarity of the inhibitor, it basically binds, it competes with the substrate, right, for the that binding site of the enzyme, right? And consequently what happens is that substrate cannot bind, and that is why the enzyme action is declined, that is inhibited, right? So basically after that we'll also see classification and nomenclature of enzymes. So basically enzymes are classes with 4 to 13 subclasses, and it is named accordingly by a 4-digit number. So here if you see, there are different types of enzymes that you see. The first one is oxidoreductase or dehydrogenase. As the name suggests, the enzymes which catalyze basically oxidoreduction, right? So the enzymes which catalyze oxidoreduction between substrate to substrates. So say there is a substrate which is reduced and there is a substrate which is going to be oxidized. Now this becomes oxidized one and this becomes reduced one, right? So this enzyme is oxidoreductase or dehydrogenase. Then there is another enzyme class of enzymes which are transferases. Now this enzyme, basically what it does is it catalyzes a transfer of group, right? Now this is other than hydrogen. So basically there are two substrates, S and S dash. Here what you see is that S is joined with a group G, and after coming in contact with that enzyme, this G, if it is attached to S, this bond is broken and a new bond is formed between S dash and G. So here you can see that now that group is basically joined with the other substrate. So these are transferases. So basically what they're doing is they are transferring the group which is attached to one substrate to the other one. Now the third one are hydrolases. Now these enzymes basically catalyze hydrolysis of water, ester. So basically hydrolysis of ester, ether, peptide, glycosidic, halide, PN bonds. So all these hydrolase bonds that you see, like peptide bond is one, glycosidic bond, we also studied about these bonds. So all these bonds, ether, ester, it's a very very important group of enzymes which catalyze hydrolysis of certain biomolecules basically. Then basically we have the fourth group, which is ligases first, right? So liases are enzymes that catalyze removal of groups. This catalyze removal of groups from substrate by mechanism which is other than hydrolysis. Remember that it removes groups other than hydrolysis, and this leaves double bonds. So say this is something like that, right? Now it removes the group. So now the group is removed, plus you have carbon-carbon double bond. So this is nothing but an example of liases. Now the next one is isomerases. So isomerases basically includes all enzyme which catalyzes interconversion of optical, geometric or positional isomers. So basically interconversion of isomers is done by isomerases. Then we have ligases. Now like we have liases which basically removes the group, here ligase is ligase, that means they link together, they add to compounds, right? So basically enzymes which catalyze joining of CO, CS, CN or PO, such kind of bonds are ligases. So guys, these are the six group of enzymes that we have to study about. Guys, the last topic is cofactors. So what are cofactors basically? Are these enzymes? Are these something else? So basically guys, enzymes are composed of what? It is composed of polypeptide chain, they are proteins, right? But there are sometimes, non-protein constituents. Sometimes there are non-protein constituents which work like enzymes, and these are known as cofactors. And what happens is they are bound to the enzyme to make that enzyme catalytically active. So say we have an enzyme, right? So this is an enzyme. Now this is inactive, but they have, say, a factor, right? So they have a non-protein, non-polypeptide cofactor, and because of this cofactor, the enzyme becomes active. The enzyme becomes active, right? So these cofactors are basically non-protein constituent present in enzymes, right? Right now what happens is basically in this case the protein portion, so what I'm saying is this portion is non-protein and this portion is protein, right? So here this protein portion is known as apoenzyme. The protein portion is known as apoenzyme, right? So guys, there are three types of cofactors which are present. The first one is prosthetic groups, right? So prosthetic groups are basically organic compounds, and they are distinguished from other cofactors as they are tightly bound to the apoenzyme, which is the protein part of the enzyme. For example, in peroxidase and catalase, which catalyzes the breakdown of hydrogen peroxide to water and oxygen, heme is the prosthetic group, and it is part of active site of enzyme. Now the second ones are coenzymes. Second one are coenzymes. So coenzymes again are organic compounds, but their association with the apoenzyme is only transient. So basically that occurs during the course of catalysis, right? Apart from that, coenzymes serve as cofactors in a number of different enzyme catalyzed reactions. Like you have coenzyme nicotinamide adenine nucleotide, which is NAD or NADP, that contains the vitamin niacin. So essential chemical components of many coenzymes are what? It is vitamins, right? So their association with the apoenzyme is transient. It is not like prosthetic groups, right? Now the third type is basically, guys, metal ions. So a number of enzymes require metal ions for their activity, and that forms coordinate bond with side chain of the active site. So even in the case of, say, zinc, which is a cofactor for proteolytic enzyme, which is carboxypeptidase, right? So many times metal ions form coordination bond with side chain of the active site, and their presence make the catalytic activity of the enzyme, being from inactive to active. So basically, guys, remember that in these cases where the enzyme needs cofactor, the catalytic activity of the enzyme is lost when the cofactor is removed from the enzyme. And this, proves that coenzymes or cofactors or, prosthetic groups or the metal ions, where they are needed, they play a very, very crucial role in the catalytic activity of the enzyme. So guys, with this we have finished the topic of enzyme as well, and with this we have finished the 9th chapter, which is biomolecules. So I hope you would have understood the entire chapter completely. Guys, do let me know if you have any doubt. You can either drop in a message in comments, or you can also message me on WhatsApp and ask your doubts. The WhatsApp number is given in the description of the video. And yes guys, if you did understand the the entire chapter with the help of my videos, please do like the videos, share it with your friends as much as you can. And yes, if you're new to the channel, please do not forget to subscribe to the channel. Okay guys, see you in the next video with a very fresh chapter. Take care. Bye bye.